So I guess now, we have time for a very quick introduction of all the group members. So I don't know if you can see what I can see. So I can see myself at the right corner, and then next to me is Matthias, and then Jim, and then Sebastian. So would you mind to briefly introduce yourself, just like I did? Starting with Matthias.
I'm Matthias Kratzler. I'm here at the University of Michigan. I'm a nephrologist and computational biologist. I'm involved with several international research networks to identify molecular mechanisms in kidney biopsy tissue and tumor nephrectomies, unaffected parts. There is most relevant here the Human Cell Atlas Seed Network for kidney. It's a kidney precision medicine project. Over the last couple of decades, we have procured tissue by various means for different profiling technologies. And I think most relevant for this discussion here now in human cell and KPNP are multicenter research networks in diseases where you get fine needle biopsies or biopsy cores only can leverage that material for maximum knowledge gain with a wide variety of technologies. And look forward to the discussion today.
Thank you, Matthias. Jim? And if not, we can maybe jump to Sebastian.
Yeah, hi, I'm Sebastian Pott from the University of Chicago. We are involved in two Human Cell Atlas projects. So one is the seed network characterizing the human heart, and the other one is the Gut Cell Atlas project focusing on ileocolonic colitis. And both of these projects usually, or mostly, involve single-cell RNA sequencing and some of the epigenomic single-cell assays. So that's, I think, for the most relevant project for this discussion will be the Gut Cell Atlas, where it's like really challenging to actually come up with good and standardized techniques for tissue preservation.
Great, thank you. So the rest of those who didn't turn on the video, you're defined as passive participants, which is great. But if you would like to introduce yourself, please feel free to do that now.
Hi, this is Jim Haygood. I'm at University of North Carolina at Chapel Hill, and I'm part of the HubMap tissue mapping center that's based at UC San Diego. And I'll be collecting lung tissue for that project.
Okay, why don't we just jump into our challenges, and let's start with the purpose of this session. So you know that we're supposed to define the HMW, how might we, ... and we have to raise the challenges that we think are worth tackling and worth discussing. And as you know, in the second part of this session, we will suggest ideas of how to tackle those challenges. So as you can see here in this green box, we just proposed a few items that we can discuss now, or we can discuss any items that you would like to suggest. And the purpose is to look at the process of tissue processing, so after the collection and on the way to the assay, and recognize what are the difficulties or challenges and name them. So someone would like to start?
Just from my understanding, the first three are discussion prompts, which we can probably start off with and then see what else emerges, right, Asaf?
Sure, yeah.
So what do you think? The first one, how is the time to preservation and processing impact your technologies? And I think, as you already indicated here, that obviously critically depends on how the preservation technologies are utilized. So is there anything else than fresh, frozen, and fixed, which we should add to that question, which certainly from my vantage point is a critical one?
Yeah, so these are the three suggestions that we thought about. It will be interesting to hear if there are any fourth or fifth option. Anyone?
I think we should perhaps add those protocols where you keep the cells or the organ or the tissue in these organ transplant buffers, where you can supposedly keep the cells for an extended period of time. We have not tried that, but it seems that some people have made good experience with that.
Yes, we have with developing human kidneys where that was a critical asset to keep the tissue viable and without minor error aspects. So very, very valid point, thanks.
Yeah, that's a great point. Any other methods?
I would like to, hi, this is Ani. I would like to add if we can discuss how we are getting the tissue, if it is from a patient or from a donor, then maybe we can discuss if it were a resection or if it was a pinch biopsy. We work with gut and the way the tissue is handled in pinch biopsy versus resected tissue is quite different. That would be very useful to talk about.
Okay, great. So
biopsies, resections, maybe even autopsy material.
Yes, that would be great.
So that's more of the sample type.
And then, you know, one option we certainly have in kidney, and I think in many other organ systems, that you also have cells shedded from the organ, meaning in kidney, urinary cells, in the lung, sputum, in the gut, epithelial cells shed, which allows you for non-invasive. Is this of relevance?
So liquid samples,
yep,
like fusion, ascites. Yeah, that's a good point. Any other sample types?
No.
We,
Sorry, go ahead, Oni.
I was going to say, if we could also talk about handling epithelium separately, because sometimes in our experience, the epithelial cells are very fragile and hard to work with. If there are any ways of tackling them separately, like do a separate dissociation of the epithelium from everything else.
Mm-hmm. Yeah, that's a very good point, and I think it's a well-known problem of everyone who try to work with the tissue, with the fresh tissue, and to kind of perform a rapid process.
I think another point that's maybe important for us to address, I don't know whether this is the best group for that, is sort of to capture actually the information, like sample information in the process of basically removing the tissue. For example, where does it come from in a given organ? Is there a good way to preserve that information, which is usually lost once you got that biopsy, or you got that, especially of large organs.
Sebastian, that's the CCF discussion which is happening in parallel here. So I think we can bounce that only if this would have direct impacts of the processing of the samples. So, in my field, we know that certain areas, if you do a tumor nephrectomy, and depending where you get the kidney out, it will be more or less ischemic and will be more sensitive to certain artifacts. And therefore, would you consider different processing depending on the source of your tissue? I think cancer with central necrotic material, I think, is a current theme there as well.
But Sebastian, did you mean to take notes of the technical steps during the process?
Well, I was actually more referring like as a well, I guess it's part of the tissue processing when you remove part of the organ to sort of be able to actually relate that information later to incorporate it into like a CCF or something like that. It's not so much the processing of, like, let's say, tissue dissociation and so on, but more like capturing metadata in a way that can be used for later analysis.
Yeah, during the process, not before.
Yeah.
And not downstream in a downstream assay like analysis, but during the process itself.
Yeah.
Yeah, I agree. It is important.
Can you see how I try to phrase that? Monitor tissue processing in relationship to tissue CCF and associated. Does that capture that, Sebastian?
Yeah, I think so.
Thanks.
Yeah. Looks good.
Excuse me, a little bit on the same topic. I was wondering, since we are talking about fragile cells, I was wondering also about adipose tissue and mainly white adipose tissue, because the cells are fairly large. So I was wondering whether there's any opinion on whether we should isolate nuclei or cells, even though I think there's a consensus there, and more to the spatial transcriptomics that I find it very hard, like whether there is a recommended thickness for the cutting so that you preserve the tissue, particularly in brown, but also in white adipose tissue. Thank you.
So is that, I would say that that's maybe more relevant to the preservation, so slightly upstream to the process. And that's maybe relevant to the first question, the time of preservation, the impact on which technology you choose to process. Would you agree?
Right, yes, thank you, yes. I would agree. It's to that more, and I don't know. Like in the spatial transcriptomics, I was wondering whether there is a minimal and maximum cutoff, like of thickness that you have knowledge that it's optimal. Let's put it like that.
Yeah, that's a good question, which I don't have the answer probably, but yeah, we can definitely tackle it in the second session.
Yes, thank you.
Yeah, and I just wonder, because it is important to characterize fat tissue, and most of the time we lose it because of the process. Did you get a chance to work with frozen tissue and try to characterize with fat?
Exactly. So far, I've been working with frozen tissue, and we have been doing single nuclei. So on the RNA sequencing, I think it's pretty clear, because there is a clear shift towards smaller adipocytes in case you're trying to go for single cells, so dissociation of single cell. But I was wondering, I have tried some sections with freshly frozen or freshly fixed adipose tissue, and the cuttings are very, very crucial, because you cannot go very thin with the thickness. The tissue breaks very easily. So I was wondering in what range I could play so that I could give it a try for FISH or RNA scope or any spatial single cell transcriptomic analysis.
So maybe we can phrase it of how to preserve and process challenging tissue like fat tissue, or I know that some people have difficulties with very fibrotic tissue when you can hardly dissociate the cells for single cell. So maybe that can be one of our challenges.
That's right.
Awesome. Any other ideas?
So one question which we have struggled a lot is if you have to procure your tissue in a diverse research network. Obviously, you have to balance on one hand, the opportunity using fresh tissue, which gives you a high integrity of the material, but with limited processing and analytical technologies present at your procurement side, versus preserving the material and sending then that to core analytical units where you have defined expertise present, but obviously the tissue has to be preserved to get there, and how to balance these and what strategies people have found to be helpful in that context.
Yes, exactly. And when it comes to human samples that they might not be, like based on the surgery time, it might not be so easy to directly process them. Based on the core, they might not be open all the time. You might need to freeze them, so that might bring a limitation.
Do you think Oh, I'm sorry, Wendy, go ahead.
I just wanted to bring up a different issue that we have had some time is once we have obtained our tissue, at what temperature do we keep it? Do we maintain them at room temperature or on ice? It seems it does make a difference, like how we are keeping or what temperature we are transferring the samples in, even if they are like processed right away. If that can be a topic of discussion, I would be very curious to hear if anyone has anything to say about not like doing a cold shock to certain samples, like transferring on ice. Do we maintain them at 37 degrees or room temperature is okay?
Yeah, that's a great point. And I think to another dimension is the time. So both temperature and time, and maybe they go together. So sometimes you can control it, but if you have a mouse that you decide to perform 20 procedures in 1 hour, sometimes you have 2 patients that are at the end of the day and by the next day. So then it's not so much up to you. So maybe we will add the timeline also to temperature. Would that sound appropriate?
Yes. Yes, that would be,
cool.
Yes, exactly.
So one point, I think it came up before, but maybe more explicitly, like we have the point prioritize processing technologies to enable optimal preservation of a specific cell type. I think one problem that, for example, Oni and I encounter a lot is sort of if you have a very heterogeneous tissue with a priori uncharacterized cell type composition, how do you actually optimize the protocol such that you both get optimal preservation of every cell type, but also like somehow like a representation that's accurate for the whole tissue, right? And I think that's a really complicated problem in some tissues where you're actually likely not able to do both at the same time.
Team, I have a problem. I'm adding my notes, but they are not visible on the Zoom any longer. Oh, there we go.
Oh, I'm sorry. Sorry, that's me.
Okay, thanks. Okay, sorry, can you just repeat? I was distracted. Yeah?
Yes, so that there is like a balance to be found between getting the for each cell type in a heterogeneous tissue to get like good quality representation versus get at each cell type represented appropriately in sort of like, let's say, proportions within the cell type.
Mm-hmm. Yeah, maybe that's part of the question of how to pick your assay, which assay do you want to perform? And also related to what was said before, maybe we should have like, you know, a short list of all the assays that are possible, and then we should have like the pros and cons or what which assay can report you about. Maybe that will be a challenge to document this. So, for example, if I want to look at CD4 cells, should I work on fresh tissue or can I freeze and then at my own time? I can work on immune cells because I can have single nuclei instead of single cells in a later point. And for immune cells, it won't be that bad to work on frozen tissue and not on the fresh option. What do you think? So in other words, should we state, or should we survey all the possible assays with pros and cons?
Yeah, and one challenge there is along the similar lines. Obviously, we know what we want to do today, but we procure tissue for the studies of tomorrow. So how can you be as much forward-looking in your tissue procurement as possible. That might be a different spin to the same question.
Mm-hmm. Yeah. So how we put it in HMW words?
Yeah, please help me.
Yeah.
I think the pros and cons of each method is a very good idea. Thank you for that. Another point that maybe we can include as a side point in the discussion would be whether during the collection of the samples, the usage of RNase inhibitors, either chemical or recombinant, makes sense, and like in what context, and like whether you have a comparison. So far, I've been using recombinant RNase inhibitors, but I don't know whether RNA later will make it better, or and maybe a side topic as well would be like freezing the tissue in liquid, let's say RNA later, or just a dry tissue in a tube, whether that makes any difference.
Mm-hmm. Yeah, so just to give you the time code, we have 10 or 15 more minutes. And I would like to suggest also to discuss the third point that was, so that's here. Whoops, I'm sorry. So that's here. The maximization of the tissue that we collect. And when we suggested this discussion point, we thought about, you know, we have a limited number of specimens. By the end of the day, if it's a mouse with 2 milligrams or 3 cores, 18 gauge, you won't be able to perform all the assays that you want. So you have to think maybe ahead of time of either share 1 core and split it to 2 assays, or just to, you want to be on the safe side and just dedicate as much material as you can dedicate to 1 assay alone.
Oh yes, we had plenty of these discussions. That will be a good one.
Anyone else had this problem or this challenge?
Yes, definitely. Working with brown adipose tissue, sometimes we get very limited amount of tissue, so that's important to prioritize.
Matthias, can you give us an example of what challenges you had around this area and what you did?
I can actually, when we go there, I can share you a flow diagram, which we refer to as a motherboard of KPMP, where out of 3 biopsy cores, we are feeding tissue into 9 different assays. And so, for example, the OCT sections each section is dedicated to a specific technology. Sounds very efficient, but comes with a lot of constraints in ensuring quality. So this is clearly one of the hardest topics if you work of very limited starting amount, as we do with protocol biopsies done from kidneys for research, how to navigate these waters. That's a really very good discussion.
And just out of curiosity, is that something that gets better with time? So you improve your protocols, then you can spare some tissue to other assays?
Oh yes, and Sanjay Jain in the QC parallel session here hopefully will describe how very tight feedback loops of quality controls are absolutely critical, so that if you have limited tissue sources, that you very early on flag when a tissue procurement protocol is not delivering, and you either adjust, optimize, or you terminate a tissue procurement strategy. Because clearly this is where a lot of the downstream utility will be determined.
So maybe that's another challenge that we can add. How do we incorporate the quality control in general in processing, but also in the aim to maximize the use of tissue? So anyone else had this problem of prioritizing or contemplate whether to, focus on one essay or divide the tissue to a few different essays?
Maybe that is something that will eventually become less of an issue as we are seeing more and more multi-omics profiling coming up, where you would hope that you would then have your core protocol, which might give you at least RNA-seq plus a tag-seq, but perhaps also some DNA information, etc.
So you think that, oh, I'm sorry, go ahead.
No, Asaf, please.
So this means we would have to balance, you have the tissue destructive protocols versus the protocols who retains the 3D structure of the tissue. And I think that's probably one of the most fundamental decisions to make. And at least right now, I see these strategies still complementary and not one entirely replacing the other. Yeah?
Can I add something? I want to point out that oftentimes tissue sample volume is very limited, but we sometimes have the other problem where we have too much tissue, and then the question becomes, how well are we sampling the different parts? So this we kind of learned from trial and error and like looking at unsatisfactory data, that it's best to sample randomly from the entire tissue region that's available. And what we end up doing, we don't do any spatial transcriptomics. I should say everything is single cell sequencing based. But we first dissociate the entire tissue and then go to different protocols, so that we have a good representation between different techniques instead of like sectioning the tissue for different protocols and then dissociate cells or nuclei, so on and so forth. If you and this question again would be very tissue type dependent. If anyone has any experiences that are similar or different, it would be very useful to hear them in this forum.
Only actually, maybe I'm not adding really to your question, but like adding another dimension. I think that what we also experience is like why a quality control is really important, sort of that we have in a way interdependent assays, and so you would like to generate some assays that are only really like useful in the context of other assays. Of course, maybe multi-omic assays solve some of that problem, but really you might be going ahead and do a bunch of assays and then realize, the second part or the tissue wasn't good enough for the second one. And so it would be useful to have actually very clear quality control guidelines that like avoid this kind of like loss of sample and also just of money, where you basically go ahead with one assay and realize later that the second one couldn't be done. And so your sample is lost in a way. So if there's like a universal QC step, universal QC step basically that gives you guidance on which genomic methods, for example, are actually possible to be done on that sample. I think that would be really useful, whether it's like realistic, I don't know.
Would an excess of tissue ever brought new collaborations or development of new assays?
Sorry, which?
Yeah, it's more of a question to Uni. Would an excess of tissue ended up with more collaborations or new essay that you thought about?
No, no, not really.
Okay, well, potentially that will be maybe an addition to the challenge that you posed. So this way we can learn more about the tissue with collaboration, or just put the tissue away, freeze down the tissue, and then
I see. Yeah, I understand your question now. I didn't catch it before.
Oh,
yeah,
sorry.
No, we definitely do store excess tissue. We bank it for sure. But again, we come up with the question, should we freeze down cells or should we freeze down entire tissue? Again, single nuclei methods are useful in many cases, but if we had a choice, do we sometimes freeze dissociated cells and in DMSO bring them up and use them like fresh cells? Or do we have a chunk of tissue that is stored in RNA later or something similar or flash frozen, and then do single nuclei dissociation directly? So the question again is like, there is some upfront decision making that we need to do. So if we do end up doing the tissue dissociation into single cells, of course, we are going to get good single cell RNA-seq or ATAC-seq data, but then we are losing the spatial information altogether. So if we want to do like, an FFPE sample to look at histology, then, we lose that opportunity. So if tissue, amount of tissue is not a problem, then maybe we can, again, divide it into two equally representative chunks and store them both as cells and tissue.
And also maybe, I don't know whether there is experience on that, but I don't know, after the isolation of nuclei, I'm not sure if anybody has directly processed it, say in 10x, or anybody has tried to freeze the isolated nuclei and in what conditions, and then recover them to again have them intact and so singularized to run them in the 10x.
Like whole cells?
Nuclei.
Oh, okay.
We have some experience on that. We usually find that doing the nuclei isolation right before running the protocol is best. We have tried freezing nuclei. So it happened that we got a bunch of nuclei. We ran some single cell assays, or single nuclei assays, and then there were leftover. And we tried running them like the week after. Usually there is a lot of compromise in data quality. Okay. So if I had the choice, I would like start from tissue and then directly get nuclei out and run the single cell assay.
Okay, thank you so much, because so far I also tried the same, so directly after single nuclei to process them, but I had frozen them down and I didn't have the chance to recover them to see how that works. So I was wondering, and may I quickly ask, in what buffer did you freeze them down? Was it BSA?
We usually freeze them not in BSA. We freeze them in FBS and FBS, media, and DMSO.
Okay, thank you so much.
Yeah.
So we have about 3 more minutes, and I would like to challenge you with another dilemma, just to follow up on what Uni mentioned now, that if you have a specimen and you want to break it into 2 parts, so would that be 2 equal parts, or you want a sample piece from each part and then mix it just to have a representation of the entire tissue?
I don't know, Asaf, this is a tough one. Are you thinking about tumors, or are you thinking about like primary tissue? Because, in primary tissue, you kind of know where you're getting what from. Okay, no, we don't know.
Sometimes you do, but in the kidney, even if you think you live in the same CCF, if you get into a medullary array, you get actually elements which are from a deeper part of the kidney than if you're in the kidney cortex. And you will know that when you disperse the tissue and you look at the cells you are pulling out, if you're in the healthy state. But if you're in a disease state where you are shifting your cellular composition as you move along the disease process, then you have 2 unknowns which are very difficult to disentangle.
Yes, yes. The converse problem is like if we were to dissociate a really large chunk of tissue and then create the most homogeneous suspension possible and sample that would be ideal, but like dissociating tissue is very expensive in terms of enzymes. So, and I think like one thing we didn't mention, but would be nice to discuss is the cold acting proteases that I just remembered. Like there are some cold acting proteases that are often brought up.
We spent a couple of years with those, and if you want to go on a journey, we can certainly help. No, this is exactly the technology. How do you balance and what works best for which cell types, and having a benchmark available. So that might be another question to add, kind of, if you do these, how do you benchmark them? And so one option is, for example, that you do a bulk analysis, yeah? Benchmark isolation strategies. Okay, talking and typing doesn't work, right?
No, that's fine.
Actually, now that you're bringing up the benchmarking, it came up in the previous breakout session as well. It's like, I mean, by now, a lot of effort has been put into some of those already, right? In other like consortia, and it's maybe not the most, but maybe it's useful to do some sort of meta analysis of actually the protocols that are already out, the benchmarking papers that are already out. Of course, oftentimes they are not really exactly reflective of the experimental system we are using in the lab, but they might help to identify sort of already like most of the like important variables.
Yeah. That you have, and this is obviously one of the essence of the Human Cell Atlas, to have exactly that atlas available, so that if you have a novel strategy, you can validate that novel strategy against what the atlas has established to be the reference.
Yeah, great point. I just want to mention that we're running out of time, so let's pick up in 25 minutes, and we can just do a quick summary for what we just discussed, and then go on to the tackle of those challenges. So I'll please take a break now, and we'll see you again in 20 minutes.
Could we talk about the first point? Time to preservation. And I'm particularly interested in the organ transplant buffer. I was wondering if it is advisable to transfer tissue or cells between labs, for example, in any of these organ transplant buffers, or should we do that in cell media, PBS, on ice, at room temperature? Yeah, like how to even bring cells into our lab. Or
what I can tell you is we have done work in that area quite extensively, using kidney transplants as a reference tissue sample. And we very quickly noticed that depending on the source of tissue in the procurement, you see significant differences depending where the tissue and how the tissue has been procured. Even if you have a living donor kidney transplant, which is a certified healthy individual under optimal condition, giving a kidney to a recipient and you remove a piece of their tissue with a protocol research biopsy, even that tissue shows a significant stress response if you compare it to a percutaneous needle biopsy obtained from a similar condition. So you have to be acutely aware that all of these preservation technologies will be significantly impacting your at least immediate early response, stress response. And the different cells will respond differently. I think that came up at multiple times when we formulated the question, that epithelial cells are exquisitely sensitive to challenges in that procurement process. whereas mesenchymal cells appear to be more resilient to these aspects. We also found that murine versus human tissues are differently sensitive, with the human tissue significantly more sensitive to these procurement artifacts. And embryonic versus adult versus aged tissue also show a sensitivity gradient. So we ended up in, giving the constraints that you have to procure tissue to have these reference data sets available. Several of them are bulk and not single cell, but at least you have them available to see if you get a stress signal emerging with one of these isolation technologies, which you otherwise don't. And conversely, if you are losing specific cell types, which you are now are present in your reference data sets with various of these isolation or preservation technologies, so that you know when you are introducing a bias there.
So maybe to that we can copy the first question, and also the first question in the second segment that starts with the scribe capture. I think it's the same. Basically, you talk about preservation and which tissue to preserve, and then we can move those to this section, to the blue section.
Yeah.
And maybe in the meantime, I can answer something that you proposed, Uni. Oh, we're here, right? So I think that there is a difference between viability of tissue, and that's the transplantation media supposed to answer, and between the integrity of RNA and the authenticity of RNA, which that's definitely a different story for preservation of tissue. That's something that at least we noticed.
To ask, it's how bad is stress response? I mean, we kind of know that there are going to be stress response. There is going to be immediate early response no matter what we do. Is it something that we can computationally factor out and then go on with our cell type classification?
I think it depends what you're asking for. If you're looking for therapy for acute kidney injury, where stress response is one of the key driving factors, and you are in a difficult spot, right? Mm-hmm. If you're looking at matrix modifications, which probably take several hours to be impacted, then you might not be. And so that, I think, reflects back what questions you are asking. And if you are primarily there to define what cell type lineages you have present, even there you have to be careful. In proximal tubular epithelial cells, depending on the tissue stress, we do see distinct subclusters, which if we use reference tissue, we see a subcluster popping up. If we expose a tissue to ischemia during procurement, which is identical to the subcluster, at least this is an N of 3 or 4 right now, but it's similar to the subcluster we see in acute kidney injury.
Wow.
And so I think being cognizant, and one aspect which I think a lot of groups are doing, that you look at these stress genes in your reference tissue procurement protocol, and if you see them compared to your bulk, then you know that that's a confounder you have present, and that you should block that element out from your further downstream analysis, so that you just know you cannot comment on those pathways in a setting of disease conditions or experimental manipulation.
Yeah, so that's a really good tip. So would you say that that's, so we should recommend the use of reference specific tissue signature? I mean, you gave a very specific example about the stress, which I think it's a really good signature. And I remember June, and there was another transcription factor that was rapidly
FOS.
changed. Right, FOS in June. Right, yeah. And that was kind of a signature to notify you that maybe your tissue spent a lot of time or was stressed out too much. So would you say that that's, yeah, I think that's a good definition. Yeah. So when you also mentioned the temperature, which we link to time, and now we can also link it to the kind of type of sample that you sample, because big resection is different than small core. Well, did you notice anything? Did you try to preserve kind of the same temperature and to avoid like freeze and 37 degrees and?
Yeah, so before we were like transferring, this is not dissociation, just transferring things from the OR to the lab. And we were doing that on ice, and we were getting pretty bad viability. And once we started transferring things in like warmed media, things got better, viability increased. We still don't have the sequencing data to do a better head-to-head comparison, but just from like, trypan staining, viability increased dramatically, transferring things on warmed media. But I almost don't want to mention this because this might be a very tissue specific thing and might not, and even for a given tissue, there might be a particular cell type that is probably more robust to, temperature shocks, and other cells not. So, yeah, just that is kind of an anecdotal thing that transferring things in warmed media definitely does help. I was actually wondering if other people. I can also tell you this is, we are working on the female reproductive system cell atlas, and this is ovary and fallopian tube that was transferred, and we saw viability with the epithelium particularly improve when we transferred things in warm media. But,
yeah, no, that's a great point.
Yeah, if it weren't the epithelium that we were interested in, maybe we wouldn't have noticed it or cared about it.
The Acceleration of Medicine program in rheumatoid arthritis and lupus nephritis, which Francis Collins mentioned, that study actually did 3.5-4 years ago a series of systematic analysis in evaluating fresh versus frozen, invarious fixative, solid tissue versus dispersed tissue, frozen, solid versus dispersed tissue in media, transferred from recruitment size to a central analytical unit. There's a paper out in bioarchives where that material has been quite comprehensively described. The first author is Anon Arasi, is from the Nature Immunology paper, which emerged out of that, and Paul Hoover, I think, was the first author of the technology paper. You will find at least a strategy how to establish these protocols and how to prioritize them as you emerge from that activity. I can probably even put that in here if it would be of help.
Thank you.
Yeah, no, it's great, and maybe the solution is to this challenge is very trivial. Just to be pointed to the right papers to read. Yeah. And actually, that's very helpful because I didn't know about this. And I'm going to run and check it out once we'll finish here. So thank you. That's a great point. Matthias, do you want to add also the temperature challenge? I think it's here at the.
Yeah, we could do that. Yeah.
So you have your own screen, right? Okay. Anything else about maybe about the sample size or sample type, whether it's liquid or solid tissue, any challenges that we can think about how to resolve now? Oh, excellent, great. How about if you're working on small tissue, let's say you're working on biopsies and now you want to switch to resections, do you have like a contact person or database that you will go there and check for available processes? You guys work with the Protocols.io?
Yeah, that's obviously a strong push from NIH-HCA that the different networks who are developing different protocols that we put them up in IO. And I'm actually not sure if they're indexed by Human Cell Atlas so that you can see, you know, which protocols are addressing or have evaluated similar challenges. Because, you know, what's so striking, if you listen to the different epithelial cell organs, the challenges are the same. But the organs behave different. So each organ needs a specific strategy to be developed for it. But the process to get there, I think there's a clear pattern emerging, which we also have seen in the discussions here over the last hour and a half. Right?
And let's say that you want to approach new tissue. Would you go straight to a human sample, or first you kind of practice on maybe an animal model?
Yeah, I think the challenge there is that we found animals to behave quite differently. For example, getting high quality single cell data from mouse kidney tissue was not a problem. Human kidney tissue behaved very differently simply due to the fact that mouse tissue usually was young mice without any fibrotic scaffolding in it, whereas, you know, human kidney tissue, when you are beyond the embryonic fetal stage, has a lot of that scaffolding, which fundamentally alters the behavior of the tissue. And the model, the protocols from the murine system were only limitedly transferable to the human, meaning we only got certain cell types viable out of human tissue with the mouse protocol, and a lot of epithelial cells were lost until the protocol was significantly altered. So as long as people are aware that you can establish the principal landmarks, but then the tissue protocols will need to be very carefully adapted to the organ, and in humans, even the age of the tissue, that what works very well with fetal tissue does not work with tissue of adults.
Yeah, I agree. Many moving parts. So can we conclude that a database reaches Protocols.io will be useful for that challenge, or did you experience any other databases, or maybe one central place that you can reach out for protocols?
I think a central place would be good, but it would be also useful. I don't think they are indexed very beyond the fact that they are that these protocols are part of like a particular consortium. It would probably be worthwhile of having this or suggesting this kind of index and potentially a little bit more metadata for the protocol that makes it possible to maybe search them. Because right now, I don't think that's the case. And the other thing is, it's one thing to have the protocols, it's the other one to have actually already performance characteristics of that protocol, right? I mean, I think a lot of times there are publications associated with some of the protocols, and it would be useful to actually see, to get an idea of how they perform, which is not obvious from our step-by-step protocol. So I think it would be useful to have that kind of information somewhat associated with it.
Yeah, that's an excellent point, and actually that's something that we can suggest to the network. Yeah. It's fairly early stage, and things can be still attached to each other. So I think that's an excellent suggestion.
Can you repeat that, please? I didn't get it, sorry.
But basically have metadata associated with the protocols that include their specific uses, but also actually publications or even like data associated with it, like performance, like very simple performance characteristics that make it easier for someone who's looking for a protocol to just see whether it's suitable for that particular context.
Yeah, I would say even just a basic quality control from the analysis can be very helpful.
Yeah.
Oh, that's a great point, Sebastian. That's great. Thanks. Yep, looks good. So anything else to add to this part? So we talked about preservation, the methods, different types of samples, the temperature and time of preservation. And we suggested, well, everything that we just discussed. Anything else to add here? Okay, so let's go back to the list above and see whether we can identify anything that we can discuss next.
I think one topic which probably for those working with human clinical samples becomes relevant, how do you decide what complexity of sample processing you do on site and versus having simple procurement protocols but allowing you more robust capture, and how do you benchmark these protocols against each other? We had that discussion earlier in the session before, if you take solid tissue, you freeze it on block, or do you partially process it? And if you partially process, how can you do that in a setting that your recruitment side, who quite often have clinical expertise, but not so much wet bench expertise, can perform that at an adequate quality standard?
So that's more on the process design or experimental design. So do you want to cut or copy and paste it here to the second box so we can start and isolate our ideas? And I bet that that's gonna, by the end of the day, also relate to this part, to the first part that we started to discuss.
Yeah.
Okay. So in other words, complexity of samples means number of essays that you want to do on a given sample.
No, sorry, I think this means complexity of sample. Yeah, it means in essence, if the extremes are, you take a tumor on block and you freeze it on block, and then that block is stored in your central biorepository, and you cut pieces of it and send it out to your sites who are doing your research. Versus the other extreme is that you take a tumor and you process it in 50 different ways of whatever potential applications you can imagine, and have it ready to go, frozen down in these different applications at the central biorepository for a research network or investigators to link up with. Those are kind of the two extremes. The first one, you can do in essence in the jungle, and that's, in our specific case, we use RNA later fixed tissue, and we literally do it in the H3 Africa network in the jungle. It's very limited impact of environmental resources and conditions, versus the other where you have in KPMP, we have 7 different individuals around the patients who is donating a kidney biopsy, who process that under kind of video monitoring in these 9 different tissue pieces for future analysis. And that obviously are the extremes, and can we provide guidance for investigators who are setting up a new study, which of these models or the multiple stages in between would be the most efficient one for them to utilize without having to go through the 3, 4, 5 years of technology developments many of us have done over the last years.
Would that be a factor of how well you can appreciate the collaborators? Maybe in other words, how to efficiently allocate your tissue. So if one collaborator needs 10 slides, that's different than, I don't know, 100 slides.
It also seems to be like a trade-off between the challenge of tissue processing at one side and the optimization versus the challenge of optimizing downstream protocols for given input, right? So you could probably argue if it's not complicated, of course you should process the tissue. But if that's not a given, is it possible that the downstream assays are actually just taking whatever they get as input? And I guess like the trade-off between the two.
Yeah, and this is, I think this is what you already see, is that, you know, with FFPE tissue obviously being so widely available, a lot of technologies are developed towards utilizing FFPE tissue. And even if it works on frozen sections, you still push towards FFPE, because obviously you gain a lot of additional access to tissue which is otherwise off limits.
I have a question.
Oh yeah, Uni, go ahead.
This is a question for Matthias. You know, I just forgot the question.
Now, feel free to email them to me. It's my last name, kratzler@umich.edu. Yeah, happy to help.
Thank you so much. Thank you.
And so when you talk about efficiently allocate tissue, so that's pretty upstream, the process. Do you plan it ahead with your collaborators? Do you plan it by yourself, and then add the collaborators and ask who needs what and how much? I don't know if you guys experienced it or.
So the question about serving an immediate need of a specific project, paper grant versus building a durable resource, is this kind of the tension here?
Yeah, on the planning step. So how did you do that, or what's your experience with that?
Yeah, it really depends. Really very much of the setting of the investigators, and obviously, you know, as Francis Collins mentioned this morning, that's where big data and efforts like Human Cell are so critical, because they are beyond the scale and scope of an investigator. Certainly, when you are setting up your lab to establish all of these infrastructures, technologies, procurement processes, so having some central units who can either provide samples or data is very helpful. And that's certainly where having these core networks in an organ-specific manner or in a disease-specific manner available can be very helpful, but quite often they are initiated and led by clinician scientists who do not have the technical expertise needed to procure biosamples in a manner that they are optimally useful for current and future technology inside.
So maybe what we can suggest for the question of how to design your tissue procurement strategy, maybe one thing can be to have kind of a round table strategy with the clinician and the pathologists and the people from the intervention radiology, so all the shareholders.
Yeah. And I think maybe, I mean, Oni, that's like a little bit on the gut cell atlas we're involved in is the maybe an iterative strategy as well. So like it turns out that occasionally, I mean, certainly as lab biologists, you miss out on constraints in the clinic. On the other hand, the clinicians are maybe not aware of assumptions that we make about the tissue that's procured, and have been really helpful on a couple of occasions to actually talk about that and just realize, okay, we're not talking about the same thing. And then having the flexibility to just adjust the protocol. I think that's extremely helpful, and not just saying we have to come up with the perfect protocol. I mean, obviously we have to come up with a good protocol, but to allow for the flexibility to go back and adjust sampling instructions, for example, which turned out to be very easy in our case. We just didn't know that that was a problem. And I think that that's really useful.
And maybe even saying that, you know, case studies where this has happened in different organ systems at a central place for new investigative communities to connect to might be helpful, right?
Yeah, that's another layer. Yeah, you're right. And Sebastian, I'm curious to know, in the gut atlas case, who kind of coordinate everything? Was that the clinician part, or maybe your part?
So this is like a physician scientist, now just scientist, Jean Cheng in our case, who was really setting up the framework for the tissue harvesting. So he's basically in contact with both us, who are just doing the basic science part, and the clinicians who are actually doing the scoping. And so the way it works is that there are people right at the operating theater, or like intervention procedure room, to take the samples. And just to get that right is sort of like important to just that these assistants know the nurse or the staff who actually provides the samples. So just having like the actually personal introductions from basically the clinicians all the way to us is really useful, and that was really instituted by a physician scientist. And I think that's in a way optimal, because you have like an overview of both the clinical side and the lab side. Because for me, for example, it's I have trouble imagining the constraints on the clinical side, and it's some things that seem like insurmountable are very, very straightforward for them, and vice versa, obviously, as well. And so his setting up was really instrumental for us, at least in that situation.
And that was, we had a similar experience in the kidney precision medicine project, where we brought together from the get-go, you know, clinician ethicists, participants, meaning people who are donating kidney tissue for the research, and then pathologists in the spectrum of technologists and molecular biologists. And really spend a full 2 years with some pilot tissue to plan the process and iterated the process on, you know, unaffected parts of tumor nephrectomies as a training material to learn how it works and establish QC materials. And then had a very rigorous process to actually train and activate sites. This is all in the public domain. If you go to kpmp.org, we established training videos, documentation, how to preserve tissue, how to optimally embed a biopsy core in OCT, how to embed in FFPE, how to preserve tissue for cell dissociations, for metabolomic analysis. And then having the technology approaches undergoing QC steps to ensure against the reference samples that they meet the quality criteria and were robust to the way the tissue was procured, meaning coming back to the earlier point that some of the technologies were developed towards the procurement protocol and not the procurement protocol solely towards the technologies, because obviously if you have, like in the gut or in the kidney, limited biopsy material to start off with, you can do only so much.
Mm-hmm.
So one aspect would be if, the Human Cell Atlas establishes just a catalog where these processes, protocols are accessible to investigators who are planning to build a new research network with sample procurement. That would be probably a helpful resource for the community, right?
Yeah, I wonder, I mean, is there anyone from NIH in this room that can answer us?
I think the gut cell atlas is represented, I think. And I think there are sort of steps involved, or like some procedures in place to actually capture that information as well.
Yeah. Also KPMP is putting all their protocols into protocol IO and on their network specific website. But that might be really something we bring forward to whoever from HubMap might want to own that. Or if there is a core unit in Human Cell who would be willing to, and this is, it's just a cataloging exercise, but cataloging, indexing, so that investigators don't have to search for extended periods of time. And the problem is, the GAD is not my research community, so I do not know which protocols are in development where before they get published either in IO or as manuscripts. So having an informal way to link the networks together and efforts, I think, across different organ systems is where everybody can win, and we can leverage the strengths of these multidisciplinary activities.
Yeah, and I think that's the aim of Protocols IO, because within the Atlas you can freely exchange protocols. So why not across atlases if we're all under the same policy of the NIH? It
exactly
seems right to me.
Exactly.
So we can either suggest that or raise it as a question if there is already an established mechanism. So just to draw your attention to this line, what additional expertise do you need? We didn't address it in the former blue box, but maybe now we can dedicate like a few seconds to think about it. What else can we bring to the table in order to make it more, to make it easier or more efficient, the allocation question? Personally, I would like to see more involvement of people from pathology or from IR that will tell us what the specimen, if it's a patient-derived specimen, what the specimen kind of worth, because they have their own process where they first allocate tissue to pathology, to clinical decision making, and then the rest will go to research. But we never know what's the kind of the best or the high quality tissue is. So I think I would personally really appreciate some involvement of a pathology assistant or interventional radiologist. What do you think about it?
Yeah.
Cool. I think in the human tumor atlas, we have quite a large number of pathologists. And so I wonder if we can wrap our brains around that question with them in the room. That would be very helpful.
And one thing which I found exceedingly helpful in KPMP was that we got our study participants engaged early on to make sure that the utility of the resource we are generating is, acceptable and motivating to them. This also gets back to the, any ethical and regulatory concerns that, what we are doing with the tissue and building these durable tissue resources is also aligned with the consent available. And this might be not the right heading here, but certainly something I would love to have if somebody starts such an activity new, kind of your cookbook, how to establish the next, gut cell atlas, or, your disease-specific network. What are key players you want to have around that table of the round table we mentioned earlier, and do not forget study participants, patients.
Yeah, these are really good ideas. Shall we go back to the green list and pick a new subject, new challenge? Are you guys up to the challenge? Okay, here we go. Maybe the tough tissue question.
Which one is that?
The
Typical cell types. Yeah, I got it. Yeah.
I guess a lot of those questions are actually not entirely discrete units, so it's hard to pick very specific ones, but that might be good and might include some of the other topics we wrote on.
Oh, so you want to go back and maybe see if they're like related subjects to that?
Well, I guess the tough tissue might include also this differential or preferential isolation of particular subtypes.
Right.
That seems to be a problem.
Yeah, you're right. So do you want to start a new blue box for that? Matthias?
Yeah, so which blue box? Difficult, this one here, right?
Yeah, maybe we can start with this one, and then if you can copy paste the question about the difficult tissue or the and then so Sebastian, you suggested to add to that the different protocols for different cell types.
Yeah, I guess that was motivated part of maybe by these kind of tissues that it's, if you have difficult to dissociate tissues, you often lose the representation.
Yes.
Yeah. Okay, cool. So what do we suggest? I think first you have to know what you're dealing with. So define what is the tissue that you're about to sample and study, and then maybe see the isolate the kind of the difficult part. So I can give an example because it's in Protocols.io, and now we also published it. So we start with the lung tissue that was really hard to digest. Sorry, and that was part of the HTEP, so the pilot project. So what we had to do is actually to dive in to see what's the problematic part. And we noticed, we should have known that because the lung is a flexible tissue that needs to expand and then go back again. There is elastin within between the cells that allows this movement. So in order to dissociate, you need to use the specific enzyme for that. That's elastase. So that's how we kind of figured out in a painful way how to approach this difficult tissue. So I wonder if you guys had the same or similar experience.
Yeah, this is particularly if you move towards disease states where you have areas of fibrosis, then if you're indeed going towards cell dispersion protocols, then these areas are particularly resilient to release. And as I mentioned earlier, we feel that the reasons that we get fetal tissue cells out so easily and adult kidney cells pose significantly more challenge probably comes from the same reason that the cell matrix interactions and the matrix composition is quite different in tissue which is still in an early developmental or physiological state where it gets more rigid and fibrotic with more advanced age or disease.
So did you have to change your protocol or to change the technology that you applied?
Yeah, actually, we learned very quickly that if you are interested in chronic interstitial fibrotic processes, that probably one technology alone will not work. So that with single nuke, you get the fibroblasts out, but you have trouble getting the associated immune cells isolated. And with single cell, it's the inverse, that you can get the immune cells adequately captured, but out of fibrotic tissue areas, you only get a smaller representation of the fibroblasts. In that specific instance, at least up to now, we still use these two technologies in parallel to see that they mutually can inform each other. And then hopefully there will be a future state where only one or an emerging technology like spatial transcriptional profiling, if you're down to a candidate set of markers which you need to profile, could be utilized.
I wanted to ask if do you use any kind of mechanical digestion for these fibrotic tissues, like the max homogenizer or something similar?
I think it also depends on your tissue source. If you use a biopsy core who is, a few hundred microns thick or millimeters thick, then you have the tissue already broken up, that a lot of your enzymatic digestions probably will be able to release a significant amount of the tissue representative cell population. If you have a big piece of solid tissue, then these strategies probably are worth considering. So that's also one point, is that understand how your tissue geometry will impact your isolation, right?
Yeah. That's exactly what I had in mind. So what we did, Uni, is that when we had this very fibrotic tissue, we used to take it, so the moment that it arrived from the OR, and we took a syringe and put it upside down. So to use the kind of, when you do this, when you press the syringe with and then with this flat area, we kind of kind of smash the tissue. But then we had to figure out the balance between a lot of dead cells and a lot of debris in the tissue, and the actual cells that we managed to get out of there by then enzymatic loss and downstream processes. But yeah, definitely that's another thing that you have to kind of optimize, and I think the challenge is not to optimize, because if you optimize on the same tissue, that's fine. But every patient, in our case, every patient is very different and every sample is very different. So again, many moving parts.
Yeah, yeah. So is fibrotic tissue is like the extracellular matrix for fibrosis something different? Like is it known what it's made of, in the sense, like could we target like collagenase A or B particularly to you know, I think like with disease it's more difficult to know what the ECM is versus regular tissue, where we can have an idea whether, you know, this space is going to work better or collagenase is going to work better. Could we make some sort of general assumptions for fibrotic tissue? And in that similar vein, could we also make similar assumptions for tissues from older donors tissue?
Yeah, that's a good point. The age, which I have no idea. What we did lately actually is to work more with pathologists and people that actually know how the tissue should look like. And then, you know, they have slides that they cannot share it with you on real time, but they can, before the case, they can kind of review the slide with you. And so, for example, at some point, hyaluronic acid became like a really important element that we had to pay attention to, and then get the enzyme that will dissociate that element. Otherwise, you know, we could have gone back and forth and didn't realize that. So I think again, the communication, the roundtable, the pre-process discussions are pretty important.
I think we had a question about fat cells, which is also interesting to me. I was wondering if we could talk about fat.
What? Oh, fat. Oh, okay.
Fat, yeah.
Yeah. No, that's
because we're talking about fibrosis. I mean, you know.
That's definitely under the difficult cell type. Definitely in our wheelhouse now.
Yeah.
Yeah. So from what we saw is that fat cells are pretty large, so they will stuck in the filter, and by the end of the process for single cells, you won't see any. Plus, they will tend to float in the tube. So once you take the soup out, the fat cells will go with them. So, and also I think that in the microfluidic device, well, maybe you're the right person to ask. There is some problem with the fatty dissociated, you know, liquid sample, because the tension, the surface tension will be different. So how do you address that?
So we have never worked with fat cells, but we work with cardiomyocytes, primary cardiomyocytes, which are very large. And, you know, we are optimizing the microfluidics using larger droplets and, you know, density matching fluids and so on to work with large cells. But fat is especially difficult because it floats, like you said. I would actually maybe use something that is more of a like a micro well or nano well based technology, like Alex's, like SQL. I would think the loading problem at least would be better solved in that aspect. So far, I have only seen people use single nuclei for fat cells. I wonder if it's really the density that is making this sample so hard to work with, or is there something else, like lipids being like inhibitory, and like no idea. I never worked with fat cells, but it is one of the things that I am interested in, because we work in the gut and there are some messenteric fat in the gut that we would eventually want to get to. Someone was talking about like brown fat and beef fat. I don't think we have the person still, but it would be, yeah, something that would be nice to discuss. I think like fat is very difficult. Neutrophils are very difficult. Epithelial cells are very difficult. And yeah, I wonder if it is something that should be addressed, like just, you know, have a technique for everything else and have a technique for fat, or, you know, still try to get one procedure that addresses all the cell types.
Maybe if the tissue is large enough, you can do several procedures and then
right,
you know, address each
right.
time. But I think lesson learned from whole cell versus the single nuclei is that with single nuclei you have more. So the libraries are actually pretty good for epithelial cells, and the reason is not that they're better. It's just that you see or you sequence less immune cells. So the bandwidth is more dedicated to the epithelial cells.
I see.
So that would be maybe one avenue. And I wonder about fat cells, because I can't recall how they look like in single nuclei.
Yeah, yeah, never worked with that ourselves, but it's definitely one in our wish list.
I think nicer maybe than having like a protocol for each single tissue. It would be nice if one would be able to sort of come up with sort of a metric that takes into account different cell specific features, and that allows one to sort of pick maybe the right approach for, you know, depending on where your desired cell type is and maybe where your other cell types are on that metric. I think that would be ideal, but it's also maybe, I mean, it seems to be more complicated and more abstract. But I think if one could come up with something like that, that would be certainly helpful in deciding on the right assay. And I have seen that for a number of, you know, just for the normal RNA, single cell RNA-seq studies, there are certainly these decision criteria that, you know, can be based on, do you go for nuclei versus whole cell? Do you use droplet versus like full length, or these kind of things? But in a similar way, depending on, like, you have fat cells, you have large cells, you have easily like lysed cells, and you have fibrotic cells. You know, what are the trait, like basically along different axes, where would they fall? I think that would be useful to have.
And that's a great idea that actually can go together with the previous idea of you know, what's the outcome of the protocols, what's the QC elements after this protocol. So
that's true,
yeah, just to go together, everything can go together with some kind of a matrix, like you suggested, that, you know, this is the tissue, these are the cells, or these are the cells that you're interested in, and that's the outcome. So like one whole thing like that will be very useful. I totally agree. Oh yeah, yeah. So it says, yeah. And to that extent, what additional expertise do you need for difficult tissues, problematic cells? Do you have any idea, like, what can make our life easier, challenging difficult tissues?
Potentially domain expertise in that particular cell type. I don't know whether that's, I mean, that's obviously always true, but like people work on fat cell differentiation, for example, for a long time. And I believe there are protocols for, I mean, we've done ChIP-seq or some other on those cells, but it's, if you're looking at in the context of the human cell atlas, obviously it's just one cell type out of many. and people with experience might be underrepresented. That might be useful, but it's also maybe not a very helpful statement.
So you could, fat cells obviously show up in most every organ in the human body. And if the gut people learn how to get them out, that would be helpful, for the lung and groups as well. So if cross organ lessons to be learned and shared, yeah. And this gets back to the protocol IO topic we had above, yeah. So index this knowledge in protocols IO, yeah.
Mm-hmm. So I wonder if there are any expertise from another field, like development or embryonic development. People that actually can materialize the composition of the cells by just knowing what's in there. What do you think?
There is a matrix biologist would be probably the most important one, but that's a dying breed, isn't it? Yeah,
Yeah, it sounds like a tough one. Well, maybe now with coronavirus, maybe no. Maybe they're back in the game.
Yeah.
How are you guys doing?
It's been very productive.
It looks like it. Very good.
Yeah. So I think now we have, any other idea, by the way, to follow up on what we just talked, or we can explore what's next. So time-wise, we have about 10 minutes. So I don't think that we can start a new challenge, but maybe we can jump back to the first one and review it again now that we suggested new ideas, and also we need to address the expertise that we didn't feel. So let me scroll it out. Yep. Yeah. So just to go back, just as a reminder, the challenges were: how's the time to preservation and processing impacts your technology? So we talked about transplantation, buffer, fixed, frozen, fresh, and tissue source that's impacting the process. So different tissue sample types, and also temperature and time. So just to go straight to the open question here, what additional expertise do you need? That kind of
one is from database design to governance entity to help to ensure indexing of the metadata into the protocols. I think this is what we have up here, right?
Yeah, so the expertise are like open resource, open database that will be more global, maybe. Is that what you meant?
Well, I meant that we need somebody who has the expertise, as I think a lot of these data are already either in the public domain or are slated as both HubMap, Human Cell, and other NIH activities are more and more committing to protocol IO. So if we would have an informatician willing to spend some time in indexing or develop an index extraction system out of protocol IO to find those, or implement, and then that gets back to the metadata standard discussions which are happening in parallel, that establishing adequate metadata standards for protocols so they become searchable would be one aspect here. So we can bring that over to the meta standard group, right?
Or maybe, do you think that we can ask protocols IO to add it as a feature, maybe?
I think that's, yeah, that's what I think the meta standard group by now is discussing. What, you know, standards do you want to have linked and tagged in the various outputs of the research networks? And
yeah, that could be very helpful.
Generally, I think protocols.io themselves, they are pretty open to suggestions. And so maybe they are actually the right people to facilitate such an indexing endeavor.
Yeah, and they can benefit from a better portfolio.
Yeah.
Yep. Yeah, okay, sounds good. So sounds like we have all the 3 boxes that we picked with more than 3 different challenges, because we kind of grouped together a few questions. I did notice that there is another question here. So the next meeting. So I don't know if you want to address that or not. What do you think?
It's not entirely clear what the format of this is going to be. I mean, somehow, given that we come up with 3 different topics, it doesn't seem to be like something that the 4 of us would do. And if every working group comes up with like several of these, I think maybe we can just say it would be useful to have these kind of this in a, like, I don't know, monthly format or something like that. And clearly for this one here, it would be more like an upfront investment as soon as possible, rather than for others where it's maybe just going along with the whole project. But
yeah, maybe, yeah, but because we have some suggestions, like cooperative suggestions, maybe we can seek for like a meeting or like a check-in in a month to see whether these were followed up or addressed. Would that make sense?
Yeah.
Okay. So, Matthias, do you think we can add here maybe a check-in in a month or something like that?
to do what in a month?
So we made few suggestions that we should follow. For example, the protocols IO feature, and I think here there are other suggestions that we can actually in a month from now check and see with the NIH people whether that was addressed, and if it kind of fell into the cracks, we can pick it up from there, and just to have some follow-up mechanism on these ideas.